Someone asked me about a year ago when we would be able to do this, and I just laughed because it sounds crazy. But turns out, through a very complicated mechanism, reverse translation is indeed possible, and even if really complicated, it is pretty cool. By reverse translation, I mean turning protein sequences into DNA sequences, the inverse of the central dogma, which could provide a nice way to use ubiquitous DNA sequencers to get information about hard-to-read amino acid-based protein sequences. But let's walk through how this method works. First, we have beads with little DNA sequences on them. Proteins to be sequenced are attached to the DNA. Then a certain molecule is attached to the protein's other end. It has a side that will react with the end of this little DNA sequence, which is partly complementary to the protein's other attached DNA. A polymerase extends the DNA sequence, thus adding molecular identifier information. Then this amino acid on the end is cleaved off. Now you have a DNA barcoded amino acid. But how do you actually encode the amino acid identity into DNA? Well, now you take an antibody that specifically binds one amino acid, and the antibody also has a DNA barcode, a piece of which is complementary to the amino acid's DNA. So once a polymerase extends from here, you have an amino acid identifier and a molecular identifier on the same DNA molecule. Extract these products each cycle and do a PCR with an additional DNA barcode, and then you have a molecular identifier, an amino acid identifier, and a cycle number or position identifier, and from there you can reconstruct the protein sequence. So, quite a lot of problems had to be solved to make this strategy work. They had to make sure DNA stayed intact during protein processing steps, not trivial. They had to make their own antibodies to bind their amino acids, and they had to figure out the chemistry to make all of their reactions very high efficiency and fidelity. So finally, testing the protocol on a very short peptide, they do manage to identify the right amino acid at each position, which is very cool, but of course at this stage, several features of the tech prevent it from being actually practical for protein sequencing. And there are some more advanced, very promising alternative methods. But I love the concept, very clever, and I'm excited to see if it gets developed further.